Okay, this is chapter 3. We're looking at cells. You notice not every cell is going to look the same in the body. Most of them are going to have a nucleus within the center, most often. And in the nucleus is going to be DNA, which is instructions for making every protein in the body. You'll notice as well, not all cells look the same. It's not that they have different DNA. It's just that those cells use only a certain amount of DNA to be that specific cell. So examples, some of the cells here, you got some fibroblasts. Most often if a cell ends in blast, it means that it's immature. But fibroblasts, these tend to make fibers to help connect other tissues together. The erythrocyte, that's just a fancy name for a red blood cell. And we'll take note that it doesn't have a nucleus. It kicks it out. It's being developed in your bone marrow. And that way it doesn't interfere with its job, which is to carry oxygen to tissues. Next picture, and the cells on the bottom are epithelial cells. These cells you tend to find making like the top layer of your skin, the inner lining of organs, should there be a cavity within the organ, and also these membranes that surround your organs, like you saw in the worksheet number 1, I believe. They're called serous membranes. You'll find those membranes are made up of epithelial cells. Some other type of cells, you got skeletal muscle for voluntary movement. We have smooth muscle. You often find within the walls of organs, pushing a solid or a liquid through the organ. For skeletal muscle, you'll notice it has a lot of nuclei per cell. And what happens is when you develop as an embryo slash fetus, you have single cells that eventually fuse into one long cell, which explains why you see many nuclei for skeletal muscle. Adipose cells, fat cells, its job is to take any extra nutrients that you have and eventually store it as lipids. And as the cell stores lipids, the fat, it pushes the nucleus off to the side. Macrophage is a type of white blood cell, and it's looking for things that are foreign so it can get rid of. Kind of like an amoeba will extend out its cell membrane to when it encounters something foreign, it'll surround it, bring in the contents, and get rid of it. And here you got a picture of a neuron, which is a nerve cell. And it has features where you have these branches called dendrites surrounding the body, where other neurons can communicate to it by releasing chemicals called neurotransmitters. And then from the cell body, you've got a tail called an axon, excuse me. And at the end of the tail, you have these little branches, they're called dendrites, where the nerve cell can communicate to targets, again, by releasing chemicals called neurotransmitters. Here you have a sperm cell. Basically, it also has a tail called the flagellum, and that's going to help propel it to eventually meet the egg. Once the female's egg is ovulated, they meet in the fallopian tube. And take note that the sperm cell in the head, it has the DNA found in chromosomes, and you have half chromosomes from mom, half from dad, and you have a total of 46 chromosomes that contain DNA. The mid piece here has organelles like mitochondria to make ATP to help propel the sperm cell to meet the egg. Okay, this is a typical picture of a cell. We're going to go over the functions of the organelles, which are their names for like organs. They're going to have specific functions. Let's start with the middle here in circular and purple. That whole purple area, that ball within the middle is called the nucleus, where you find the DNA, which are instructions to make proteins. And the DNA is found within structures called chromosomes. And so when the chromosomes are not as condensed, they form what's called chromatin. And then when the cell gets ready to replicate, it'll condense down to form chromosomes. In the middle of the nucleus, called nucleolus, that's where cells will make ribosomal RNA and transfer RNA, which are going to be involved in making a protein. these ribosome RNA, transfer RNA, and messenger RNA, they can eventually leave the nucleus through these little openings called nuclear pores, and where the messenger RNA can be read to make a protein. Continuous or attached to the nucleus, which you have in this blue here, is called the rough endoplasmic reticulum. And it's considered rough because it's embedded with these ribosomal RNAs. And typically when you make a protein, if you need to modify it by adding sugars, it's going to be taken to the rough ER to modify the protein. After the protein's modified, it can get packaged and shipped in a vesicle to the next organelle here in green. That's called the Golgi apparatus. And that Golgi apparatus is going to package the protein, and then it's going to ship it off to its final destination. So one of the final destinations may be to excrete something out of the cell. So in this instance, you'll see a vesicle containing the protein to be excreted out of the cell. Another function may be to replace proteins in the cell membrane, maybe replace proteins in the cytosol, which is a liquid matrix that all the organelles are suspended in. And now that I mentioned cytosol, the liquid matrix inside the cell, try not to get that confused with cytoplasm. Cytoplasm includes not just the liquid matrix of the inside of the cell, but also the organelles that are suspended in the cytosol. We've seen this organelle before in chapter 1. Kind of looks like a bean structure. It's called mitochondria. And the mitochondria are typically where you make the majority of your ATP, that molecule that all cells need to produce in order to survive. Next to the mitochondria, you have these two yellow structures. They're called centrioles. Typically, some cells can copy themselves. And when you copy the chromosomes, we need to separate them into two new cells. So those centrioles are necessary for cell replication to separate chromosomes. And process of a cell copying itself is called mitosis. And the purpose for mitosis is for growth or repair. Let's jump to the organelle here in blue called the smooth endoplasmic reticulum. Well, it's considered smooth because it's not embedded with ribosomes. And the function there, the smooth endoplasmic reticulum, is to make steroids, also to break down toxins. And steroids, basically, if you recall, we're going to use cholesterol to make steroids. These little lines within the cytosol, they're called in general, the cytoskeleton. I like to think of them like kind of like a railroad system where like tracks is going to allow, in this case, organelles to get from one area to another. They also, similar to our own skeletal system, give the framework inside the cell, giving it structure. Lysosomes, to the side of the bottom, well, top left. This lysosome is a little vesicle that contains digestive enzymes. So that way, whenever the cell wants to bring something within it, the lysosome can fuse its vesicle to what was brought in, and those enzymes will break down what was brought into the cell. And peroxisomes, down to the far bottom right. Peroxisomes, they contain enzymes to get rid of what's called free radicals, which are basically waste products that can cause damage because they're lacking an electron. If you recollect in chapter 2, things like to pair up electrons in their outermost shell to where the shell is full. So if you have chemical reactions and you form a waste product that's lacking electrons, it can pull from other areas, causing damage. They're referred to as free radicals. And therefore, peroxisomes, they have enzymes to counteract the free radical. Well, let's go to the next slide. Oh, we're looking at the cell membrane. And if you recollect from the video of chapter 2, the major component of all our cell membranes is a bilayer of phospholipids. So these phospholipids, little sphere regions, you're going to have a region that's polar, which will interact with water. So therefore, one side of the phospholipids face the water environment outside the cell, and the other phospholipids, the little round polar region, they're going to face the water environment inside the cell. The tails, we say, are nonpolar. They're hydrophobic. They don't want to mix with water. So they're not facing the water. They face the other membrane's tail, which is nonpolar. Now, you can have proteins embedded within the cell membrane, and that's what you see here. And they have a variety of different functions, which we'll look over probably in the next slide. I just want to bring to your attention that the portion of the protein that's embedded within the tail region, it's nonpolar. If it's exposed to a water type environment, that part of the protein would be polar. Also in the membrane, you see the cholesterol. I know cholesterol we use to make steroids, but in this case can help to hold the cell membrane intact, so that way it doesn't come apart. You'll also have here in green attached to the membrane sugars, and they'll have a variety of functions as well, maybe for like cell recognition. You also have proteins inside the cell, like the cytoskeleton, so give framework to the cell, allow organelles to get from one area to another, but also if it's attached to the membrane, can also help hold the membrane intact, so that way it doesn't come apart. Okay, so if you have these proteins embedded in the membrane, what could possibly be their function? Well, it may be a channel just to allow contents in or out. And as you can see here on the channel on the left, things just naturally want to go from high to low. But if you're trying to push a substance from low to high, you need energy in the form of ATP. But again, one of the functions of proteins embedded in the membrane may be a channel. It may be what we call in generic a receptor region, where a chemical will bind to this receptor protein, and that'll activate some sort of cascade to have some function involved in homeostasis. So, like, give you an example, neurons, nerve cells, they communicate to targets by releasing chemicals. Well, these chemicals do bind to a receptor protein before they get the response at the target. Proteins in the membrane may also bind to proteins either outside the cell membrane or inside, and that will help keep the membrane intact. That may be an alternate function of a protein in the membrane. Or it may be an enzyme. You have enzymes free floating around in the cytosol of the cell, also in organelles, but it looks like in this case, it may also be embedded in the cell membrane. And recollect, enzymes help speed up chemical reactions. And again, enzymes are proteins, but not necessarily every protein is an enzyme. Some proteins, they just may bind cells together, okay? Think of maybe in the stomach where it's very acidic. You don't want acidity seeping in between the cells. So between the cells, they're tightly knit with proteins. If you see a sugar, in this case where it's green, it's representing a sugar attached to a protein. It may serve for cells so that they recognize each other. That may be one of the functions of a protein. Now, these proteins that do bind cells together, let me take a closer look at each one. If they're found in high amount and yet they're kind of small and they're the majority of the ones found to knit the cells together, those are referred to as tight junctions. The other thing I want to bring to your attention here is you see the cell membrane here, it's going up, down on top. That is called microvilli. And anytime you see microvilli, which is extensions of the cell membrane, that's to have the cell doing better at absorption. So let's give an example, like in the small intestine, where you absorb nutrients. When you get to the cell level, you're going to see microvilli on the cells. Okay, now if you were to find a protein and it's not that many, it's kind of like a main large anchor between the cells. Those are going to be referred to as desmosomes. And again, those you find in between cells are large but not as abundant. And then last but not least, you may have some proteins between cells, just to kind of like a doorway between two rooms. And that's going to allow contents from one cell to quickly go to the next cell. Those type of proteins are referred to as gap junctions. One of the things you're going to be doing in lab is looking and trying to understand the term simple diffusion. So in this case, you got a beaker of water, and let's say you're going to add like a dye, or in this case, this pellet, this substance, and it just simply wants to disperse, move away from where there's a lot of to where there's little until it's evenly dispersed in the liquid. So there's a term called simple diffusion, which is the movement of a substance from high to low concentration. In other words, it moves where there's a lot of to where there's less of. And since we're referring to simple diffusion, let me just bring to your attention, I'm going to draw kind of like a hill. And let's say in the bottom of the hill is down low, and then up top is high. So anytime you see a substance just naturally want to go where there's a lot of to where there's less, think of going downhill. You don't need any energy. So that type of movement, transport, it's referred to as passive transport. It means doesn't require ATP to do so. And one type of passive transport is the simple diffusion you're seeing here. And simple diffusion, again, is the movement of substance from high to low concentration. Simple, and then I don't want to have to spell it all out just for time's sake. I'll just abbreviate it. Now, anytime you're trying to push a substance where there's less of and trying to force it to where there's more of, you're going to need energy in the form of ATP. And that type of transport where you need ATP, that's going to be referred to as active transport. And I believe the next slides are going to go over the different types of passive transport. So we've gone over one right here. It's called simple diffusion. Again, the movement of a substance from high to low concentration. Okay, let's see if I can get rid of my drawings before we go on to the next slide. All right, so let me just take these one at a time. Let's look at the far left one. So here it is magnified. Here's our cell membrane, which is a bilayer of phospholipids. Get used to the term that our cell membranes are selectively permeable, or you could say semi-permeable, which means they'll allow certain things through, but not everything. Preferably, if something's allowed through, it's small. And in order to get through the cell membrane, it has to be lipid-like, which is non-polar. So if you recollect, here's our bilayer of phospholipids that make up the cell membrane. The tails, they're non-polar, they're lipid-like. So this substance you see wanting to come in, first of all, it has to want to come in if you're not going to need energy. So it wants to come in where there's more of, high, and wants to get through the membrane where there's less of, from high to low concentration. So first criteria wants to want to come in from high to low. And then the next criteria to penetrate the cell membrane, it has to be nonpolar, lipid-like, to get by these tails in the middle here. And that's, again, if you recollect the term, the definition is called simple diffusion. All right, let's look at the next slide. All right, we're looking at a substance here. Again, there's more of it outside the cell. So it wants to come in from high to low. Right, so no energy is going to be needed. However, the substance here happens to take the shape of the channel that's going to allow it through. And it didn't penetrate the cell membrane. Remember, if something's easily going to penetrate the cell membrane, It has to be lipid-like, non-polar, just like the tails of the phospholipids. So we look at the substance, it's saying it's lipid non-soluble, which means that this substance, that substance is polar. There's a charge to it. So that means it cannot easily penetrate the cell membrane. However, it can be helped across, and if it's going to be helped across from high to low and no energy was needed, because it's helped across, this is called facilitated diffusion. That's the way I'd hope you learn it as facilitated. Yes, it says carrier mediated, just indicating it took the shape of the protein channel to be allowed through. But again, focus on facilitated diffusion. So in this next scenario, again, you have a substance. There's more of it outside the cell, right? And it naturally just wants to go where there's less of it, from high to low amount. But you'll notice the substance again is polar. It is not it's not like a lipid. So if it's polar like, the only way it's going to be allowed through is going to be helped across again by a protein channel. And that's why I recommend you learn it as facilitated diffusion. Just like in this example and the previous one, it's a type of facilitated diffusion. It's just that the substance here didn't have to take the exact shape of the channel. It's just easily allowed through. All right, let me clear these out, the drawings, or I can go to my next slide. So the previous scenario is one type of facilitated diffusion. The substance was helped across, but no energy was needed. So that's called facilitated diffusion. Same scenario here. Substance was helped across, couldn't penetrate the cell membrane, but it's still from high to low concentration, indicating that no energy was needed. And then the third type of passive transport is specifically the movement of water from high to low concentration. Now, typically water is polar, right? So water does not easily penetrate our cell membranes. However, water is small enough where some molecules may, over time, squeeze their way through because it's tiny. But usually polar substances don't penetrate our cell membrane. Water more often is allowed through channels called aquaporins. And this picture is just showing the movement of water from high to low concentration, from where there's a lot of to where there's little. Again, no energy is needed. So this type of passive transport is referred to as osmosis. Clear the drawings. And let's just review real quick. So there's different types of passive transport. And in other words, you don't need ATP, as long as the substance is going from where there's more of to where there's less of. So the movement of substance from high to low concentration, the term's called simple diffusion. The movement of a substance from high to low concentration, but it's been helped across through a channel. We're going to refer to that as facilitated diffusion. And then specifically, if it's the movement of water from high to low concentration, that's going to be referred to as osmosis. All right, let's take a look at this scenario. Let's just take one picture at a time. Take the far left scenario. In red are solutes, and in blue is solvent, the water. And if things just naturally want to go from high to low, well, let's have a look here. There's more water here on the left side, so that means it wants to go to the right. And because there's more solute on the right, it wants to go to the left until everything is equal, so that you have the same amount of solvent to solute ratio on the same side. Now, notice there is a membrane here between the two sides of this container, and that membrane is large enough to allow both the water and the solutes to move until everything is equal. So what we're looking here is if the solute moves from high to low concentration, that was simple diffusion. And then for the water to move from high to low, that's referred to as osmosis. All right, let's go look at the next scenario. Now we have almost the same scenario. The only difference here is that this membrane between the two containers, this membrane's only going to allow water to go through. The solutes are going to be too large. Okay. So again, there's more water on the left. It wants to go to the right. Okay. Now, you may not have equal amounts of solvent to solute on each side here of the containers, but something is similar here. So let me kind of draw this out. Let's say if you have solutes to solvent is 2/4. I know that looks like a 1. I'm trying to draw 2. If I could reduce that, that would be what? 1/2. So you may not have equal solute to solvents on both sides of this container here, but the ratio of solute to solvent is the same. So that's what's being balanced here. And again, let me clear the drawings. What you have here is the movement of a substance from high to low, that's simple diffusion, and specifically the movement of water from high to low is osmosis. And this membrane is representing that our cell membranes are selectively permeable, which means they don't just allow anything through. All right, this is a scenario where if you're given in solutions in the body, you want to mimic what's in your cells. That way it doesn't draw water out of your cells in excess, nor will excess amount of water want to go in the cells. You want to, if you introduce solutions in the body, to mimic already what's in the cell. So let's just take each scenario one at a time. So solutions where you want them to mimic what's already in the body are referred to as isotonic. Isos means the same amount of solutes in comparison to the cell. Iso means same. And then tonicity, tonic, means solutes. So let's just say you have a beaker of water. You draw the beaker. And you have decimal 0.2% solutes. And then the rest is water. And in the cell, you have the same amount of solutes, 0.2%. That means if you were to immerse cells in an isotonic solution, if one water molecule moves out, one water molecule will move in to counterbalance the movement. And the cell, in this case, it's a red blood cell, the shape would stay the same, which in this case, it's biconcave. Okay, so again, when you want to introduce solutions in the body, you want them to mimic what's already in your cells. So no extra water in excess goes in your cells or comes out. And solutions like that are called isotonic. All right, let's clear the drawings. Let's go to the next slide. Now, what would happen if you put your cells in what's called a hypertonic solution? Well, that's similar to kind of like putting salty water in the soil of a plant. That's going to draw water out. So hyper is referring to more of, let's try to spell this out with a mouse. I know it's not pretty. And then tonic, again, referring to the amount of solutes. So this solution that I draw in the beaker, let's say it's hypertonic. Let's say it has 10% salts. That's a lot higher than 0.2% solutes in the cell. So 10% solutes, I know I said solutes, same pretty much. And then in the cell, it's 0.2% solutes. So that means there's more solutes in the solution than in the cell, and water is going to be attracted to go where there's more solutes. So this would draw water out of the cell, going where there's more solutes, and the cell would shrink. And the term we use for shrink is we say crenates. So yeah, you want to avoid introducing in the body hypertonic solutions. That would cause our cells to crenate, which is to shrink. All right, erase this. Go to the next scenario. And what would happen in your cells if you were to introduce a hypotonic solution? The prefix hypo means less of, so a solution that would have less solutes in comparison to what's in your cell. All right. Well, let's just say this container of water has no solutes. It's all water, 100% water, no solutes. The molecular formula for water, recollect, is H2O. So that means that typically there's more solutes inside the cell, which would be 0.2, even though it's a small amount of solutes. There's still more solutes in the cell. So when you put the cell in a hypotonic solution, water is going to want to go in the cell, causing it to expand. And since there's a big difference, the cell may burst. And the process of it bursting, we say it lyse. So you want to avoid hypotonic solutions just as well. It may cause your cells to inflate, and there's a big difference. It may even lyse. And clear the drawings, and one of the experiments you'll be asked if it's done in your lab is to immerse your own cells, one in an isotonic solution, the other in a hypertonic, and then hypotonic on a slide where you can look at it underneath the microscope. So that way you can see how the cell shape is affected. All right. Okay, this picture here is showing us that all our cells pretty much have what's called a sodium-potassium pump, where this pump is going to pump out 3 sodiums. And let me get the marker out. And notice there's more sodium outside the cell. So if I'm trying to push something from low, that should have been O, from low to high, in this case, the sodium, I need energy in the form of what? ATP. And that's where you see it here. So pumping the sodium out here required energy. And then simultaneously, 2 potassiums are going to want to come in. Okay. So the energy is expended in this first scenario. And anytime you need energy to move a substance from low to high, it's called active transport. Now, if you look over here, because you establish what's called a concentration gradient, in other words, you have more ions on one side of the cell, sodium, and you have less of it on the other side of the cell. So there's more sodium outside. Therefore, if you have a channel that's just naturally open, sodium wants to come in. No energy is expended there. Now, the potassium, you establish a concentration gradient where there's more K plus potassium inside the cell in the first scenario where you needed ATP. But again, if you just have a channel that's just naturally open to allow it in, it wants to go from high to low. In this case, would go out of the cell. So typically the scenario for number 2, you didn't spend any energy here. So it's really not active transport. The actual active transport occurred in scenario number 1. So when you create a concentration gradient, you can have ions, in this case, sodium, potassium, just naturally go down their concentration gradient from high to low in this scenario number 2. And they refer to that as secondary active transport because no energy is expended here. It was actually expended in scenario number 1. All right, what are we looking at here? Well, we're looking here when anytime a cell takes contents within it, it's called in general, it's called endocytosis. Endo means within, cyto is referring to the cell. So endocytosis, the cell's bringing contents within it. And there's typically 2 types of endocytosis. Here you're going to see the cell membrane kind of fold in on itself to bring the contents in. When you see the membrane fold in and then eventually pinch off and form a vesicle, the contents were brought in through what's called pinocytosis. And we'll see that probably in the next slide. So let's say these contents were something that the cell wants to break down. Well, here's our lysosome that contains digestive enzymes. It'll fuse with the vesicle that brought the contents within it. Therefore, the lysosome's enzymes can break the contents apart. All right, let me erase the drawings. Now, anytime a cell excretes something out, like in the bottom scenario, letter B, that's going to be referred to as exocytosis. Think of exo as exiting. So that's exo. That should have been E. That should look like a C, but it doesn't quite. It's like my hand's getting tired on this mouse. So again, that's exocytosis when contents are leaving the cell. All right. I did mention there were two types of endocytosis. Let's just take one at a time. So here, kind of like an amoeba, where amoebas kind of extend their cell membrane out to bring contents within it, that is called phagocytosis. So when the membrane extends outward to bring contents within it, they form these false-looking feet structures. They refer to them as pseudopods. So on each side here, it's forming pseudopods, and then it's going to close in, forming a vesicle, just like the cell membrane, mainly of bilayer phospholipids. But the main thing here, this is a type of endocytosis called phagocytosis. The membrane folds outward to bring the contents within it. And usually, typically the substance is going to be something large and solid-like in order for phagocytosis to occur. Now, the other scenario, another type of endocytosis is called pinocytosis. So in this scenario, instead of surrounding the contents by forming pseudopods like we saw in the other scenario, the membrane is going to fold within itself to bring the contents in and then close off the phospholipids to form this vesicle. And typically, usually the substance is going to be small, liquid-like, and that's when the cell would do what's called pinocytosis. Okay, again, there's two types of endocytosis. The substance large, solid-like, the membrane folds out. That's called phagocytosis. The substance is small, liquid-like. Typically, the membrane folds in on itself to bring the contents in. That's referred to as pinocytosis. All right, last scenario. Typically, this is pinocytosis as well, but just to know when would a cell know when to take the contents within it. Typically, the cell has receptors, these proteins in the membrane. And let me get the marker out. So here's the substance, and it fits perfect on the protein, indicating to the cell, hey, this is something we want to take within ourselves. So that's when the membrane is going to fold in. It is pinocytosis, but more specifically because the content's bounded to a protein receptor, telling the cell, hey, take this in. You can also refer to that as receptor-mediated endocytosis. What I need you students to focus on are just pinocytosis, substance small, liquid-like, membrane folds in, versus phagocytosis, the substance is large, solid-like, the membrane folds out to bring the contents in. All right, let's look at this scenario here. Let me just get to the picture. So let's say the cell made something, right? The instructions to make proteins are in the cell's nucleus, the DNA. So you made something, and now the protein, in this case, is going to be excreted out of the cell. Well, how does they know when you make a protein? How does it know to be excreted out exactly? In other words, how does it know where to go? Well, here you got a vesicle with the protein that's going to be excreted out of the cell, and then you have a protein embedded in the membrane. So that this protein is going to bind to proteins from this vesicle to the protein in the membrane, and kind of like a key to a lot, they have to fit perfect together. And that typically is how it's guided to where it needs to go. And those proteins, they fuse together, and you have a bilayer of phospholipids in the vesicle, and those phospholipids are eventually going to fuse and incorporate themselves into the phospholipids of the cell membrane. And once that occurs, the contents are excreted out, and the term is called exocytosis. An actual electron microscope, of exocytosis. And this bottom scenario, this is 1,000,000 times magnified. All right, what are we looking at here? Well, let's have a look. Typically in your cell membranes, yes, we did mention you have what's called a sodium-potassium pump that would establish that there's more sodium outside the cell and there's more potassium inside the cell. So because there's more potassium, let's say inside, if there were a channel that's just naturally open called leaky channels, it wants to go out from high to low concentration. In other words, simple diffusion. You don't need any energy. Hey, what's going on here? Why is this potassium wanting to come back in this other channel? Okay, ions. If you recollect in chapter 2, ion is anything with a charge. So positive ions like sodium here, they're more specifically referred to as cations. And if you have a negative ion, they're referred to as anions. If you count all the ions inside the cell, both positive and negative, you'd have the same amount. If you count all the ions outside the cell, both positive and negative, you'd have the same amount. However, when you compare the amount of ions outside the cell versus inside, it's more electrically negative. That doesn't mean you don't have any positive ions. You can see here potassium has a positive charge. It's just saying when you compare the outside to the inside's more electrically negative. Therefore, that's why they put the A here for more negative, anion. So in this first scenario where potassium leaves, it's going down its concentration gradient. In other words, it's going where there's much of it to where there's less of. Therefore, that's simple diffusion. And then what's attracting it to come back in, if you recollect, opposite charges should attract. And we said the inside's more electrically negative compared to the outside. So at some point, the positive charge will also cause the potassium to want to come in. So therefore, one leaves, one counterbalances and comes in the cell. So this is illustrating basically that all cells obey their concentration gradients where they want to go from high to low amount. That's scenario number 1. But they also have to obey their electrochemical gradient, which is their attraction to where it's more opposite in charge. All right, here's our picture of the mitochondria. Let's just reiterate, they make ATP in high amounts. All cells need to make ATP to survive. Something special here about mitochondria that we don't see in other cells. Typically, any instructions to make proteins that the cell needs is located in the cell's nucleus. And the instructions to make proteins in the cell's nucleus is DNA. However, if you look at the mitochondria, it has its own DNA to replicate. So no other cell or any organelle has their, no other organelle has their own DNA for replication except the mitochondria. All the other DNA within the cell should be in the cell's nucleus. And remember, DNA is to make proteins. So in this case, mitochondrial DNA is for replication of mitochondria. Well, how does this happen? Well, the theory is back in the day, which means not necessarily true, they had to come up with some type of explanation. The theory is that you had 2 separate organisms eventually coexisting as one. So that's how they try to explain that mitochondria, that's why they have their own DNA. Clear that. Let's go to the next slide. Oh, that's just a larger picture of the mitochondria. It has an outer cell membrane and an inner cell membrane as well. And then it has enzymes in the inner membrane to help make ATP. There's the picture of the enzymes. An actual electron microscope image of mitochondria. The other unique thing about mitochondria is that you get it from mom. If you can recollect the picture of the sperm cell we saw in the very beginning of the chapter, the head's the only thing that's allowed into the egg. And in the head is just chromosomes. All the organelles when the sperm is being produced was kicked out. So that means the mitochondrial DNA or organelle, you get that from mom's egg. Excuse me. All right, we're looking at a picture of the rough endoplasmic reticulum down here. It's considered rough because it's embedded with ribosomes. And the function here, the rough endoplasmic reticulum, is to modify proteins by like adding sugars. And then to focus on the smooth endoplasmic reticulum, the smooth ER is smooth because it's not embedded with ribosomes. And the function of the smooth ER is to, one, we said make steroids derived from cholesterol, and also to break down harmful toxins. Just looks like to be an electron micrograph of the rough endoplasmic reticulum. And this is the Golgi apparatus. So immediately after you modify the protein at the rough endoplasmic reticulum, so it's coming from the rough ER and it's encased in a vesicle and it's shipped to the Golgi apparatus. This organelle in green has a receiving side for the contents leaving the rough ER. The receiving side is called the cis. Then the protein's re-encased in a new vesicle that's going to guide it to its final destination. And the shipping side here is called the transphase. So the final product within this vesicle leaving the Golgi apparatus, it may be some content that is excreted out of the cell. It may be to replace proteins embedded in the membrane, or maybe it's a new organelle to replace old organelles inside the cell. All right, we saw this picture. There's the electron micrograph of the Golgi apparatus. So again, what are some possible destinations once the protein is made? Something excreted out of the cell, maybe incorporate to replace proteins embedded in the membrane, or to replace organelles inside the cell. And now they're going to focus on the organelle called the lysosome. So lysosomes, they contain digestive enzymes. Therefore, when the cell brings something within, and if it needs to be broken apart, the lysosome will release enzymes onto what was brought in to break it down. And we're back at our typical picture of the cell that we saw in the beginning. Let's just look at the cytoskeleton. Let me jump back to this picture. So the cytoskeleton, you see these lines here. They're made of proteins, okay? And the cytoskeleton, they give shape to the cell. And they also kind of like a railroad system. They're going to allow organelles to get from one area to the other. And let me clear these drawings. So the cytoskeleton consists of proteins called microfilaments, like in letter A, letter B, intermediate filaments, and also as well microtubules. They're the different types of protein that make up the cytoskeleton. Now, you may see some microtubules also making up these organelles called centrioles. And again, centrioles are necessary for the cell to replicate itself. They're gonna separate chromosomes when they're copied into two new cells. Larger picture here of the centrioles made up of microtubules. Imagine that's an electron micrograph of the centrioles. Now, microtubules can also be needed in order to make what's called cilia. Cilia are proteins, kind of like hair-like projections, like in your trachea, that are going to help propel mucus. Okay. So in this case, try to imagine the lining of your trachea, your windpipe, And on the cell membranes is cilia, protein extensions, and their job is to like propel mucus. So you see these cilia doing kind of like a wave-like movement, so that way any mucus can move. Please don't get cilia confused with microvilli. Microvilli are extensions of the cell membrane, which is a bilayer of phospholipids. So when you got a cell membrane, usually you typically draw a cell circular, but when you've got the cell membrane, instead of just making a circle, it's going up, down, up, down, up, down, you're increasing the amount of surface of the cell. And when you increase the amount of surface, it makes the cell better at absorption. So think of like a napkin. The more surface the napkin has, the better it's going to absorb. So where do you see microvilli? Okay, well, we're going to see microvilli where we do most absorption of our nutrients. So that would be in the small intestine. Most digestion and absorption occurs in the small intestine. It looks like we're going back to the beginning. They're going to focus here on the nucleus, and recollect the nucleus consists of chromosomes, and if the chromosomes are uncondensed, it's referred to as chromatin. And that chromatin contains the instructions to make proteins called DNA. All right, let's get to the main picture here. So here we got chromatin, and then it starts to condense on itself. And then eventually you see this little X pattern. There's your chromosome. So this chromatin, they start wrapping around these proteins called histones. Here in purple is the chromatin, which is DNA. And it starts wrapping around these proteins called histones, and it starts to condense. And when it condenses, you can physically see now the chromosome. Now, what you're looking at here is not just, well, yes, typically it is one chromosome, but one of these strands is the original, and the other strand is its copy. So when you have the original attached to the copy, there you can refer to each one here as its sister chromatid. And typically, when are we going to see DNA copied? Well, that's when the cell is getting ready to copy itself, and it's going to separate into two new cells. And the purpose of that is for growth or repair. And the organelle, if you can recollect, what separates out the chromosomes is centrioles. So again, we're looking at the chromatin condensing down to form chromosomes. And once they condense down, you have the original and the copy. They're referred to as each other's sister chromatids. All right, here we go. The process of the cell copying itself, it's called mitosis. But the purpose, if you're asked what's the purpose of mitosis, the purpose is growth and repair. So before a cell separates into two new cells, it's got to go through a stage where all the organelles and the DNA, which is found in chromosomes, is all copied. So this period of growth where everything's copied, okay, I just need you to know it as interphase, okay? Yes, interphase gets broken down in the G1 substage where it says it's growing. The S phase where it looks like what? DNA is being copied. And then the G2 phase where making final preparations to separate everything that was copied. What you want to focus on, the growth stage where everything's copied, both organelles and DNA, that is referred to as interphase. Now, once everything's copied, we're going to go ahead and start separating the contents, the chromosomes. So that's when you go through here what's called mitosis. And the substages of mitosis in order is prophase, metaphase, anaphase, and telophase. Cytokinesis typically isn't part of mitosis. It's the end stage of it. Cytokinesis means the movement of the cells into two new cells, right? You need to separate cyto, cell, kinesis, movement. All right, let's focus on the substages of mitosis now. Let me get to the other slides. Okay, we'll come back to this slide in a bit. They're just a little bit out of order. We're looking for substages of mitosis, all right? Here we go. So we'll take one scenario at a time. The cell's going through interphase. What's going on in interphase? Everything is being copied. All the organelles, so you can see here centrioles are being copied, as well as the chromosomes. They'll start to condense down to form chromosomes. Chromatin will condense down to form chromosomes. So interphase is a stage of growth. Okay, prophase. Well, if you look here in the pictures, you got early prophase, late prophase. I need something to focus on so I know I'm in prophase. What I typically tell students, as well as I was told as a student, is that's the first time you see chromosomes. You know you're in prophase. Before, it wasn't chromosomes, it was uncondensed. It was called chromatin. So the first time you see chromosomes form, you know you're in prophase. Granted, yes, centrioles are making their sides to one side of the cell, the other is making its way to the other side of the cell. Yes, the nuclear envelope's being broken apart, it's degenerating, and centrioles are forming their proteins called spindle fibers to attach to the chromosomes to eventually separate them. But that's a lot of things to remember. So to make it easy, you know you're in prophase when you see chromosomes form for the very first time. All right, after prophase, the next stage is called metaphase. The simplest way is meta is referring to the middle. So you'll notice all the chromosomes are lined up in the middle. You know you're in metaphase when that occurs. And then anaphase. Well, these centrioles here, they're attached to the chromosomes. Now they're separating out the sister chromatids. So when you separate out the chromosomes and you see space between the chromosomes, once you see space between the chromosomes when they're being separated, you know you're in anaphase, separation of the chromosomes. All right, the last stage of mitosis is called telophase. How do I know I'm in telophase? Well, that's when you see a cleavage furrow, this little indentation form between the cells that are trying to pull away from each other. So you know you're in telophase when you see a cleavage furrow form. Cleavage furrow. And then not really part of mitosis, just the physical movement of contents going to one cell and the other one go to the next in equal amounts. That's called cytokinesis. Cyto means movement. I'm sorry, cyto means cell. Kinesis is the movement. And if you're asked again, what's the purpose of mitosis? Please don't put copying itself. The purpose of mitosis is for growing or for repair. So kind of review the stages. Prior to mitosis, everything's copied. That is interphase. When you see chromosomes form for the very first time, you're in prophase. When chromosomes line up in the middle, you're in metaphase. Separation of the chromosomes, you know you're in anaphase. And then when you see a cleavage furrow between the two cells forming, that is telophase. And not mitosis, but at the end, separating into two new cells, the contents being separated is referred to as cytokinesis. All right, let's jump back to some of these slides that kind of missed out on. Okay, so here you're looking at the DNA inside the cell. When DNA is copied, what typically occurs is you're going to have an enzyme. In this case, it's called DNA polymerase, and DNA is double-stranded. And the polymerase will unwind this one of the strands, and it'll make its complementary strand. And then on the other end, you have a DNA polymerase making the complementary strand as well. And when you get the two new strands, they each have a piece of the original strand. So often when DNA is copied, the chromosomes is referred to as semi-conservative replication because it's been conserved that you use the original strand in each new DNA. Now let's have a look here. DNA is double-stranded. It's in helical format, which means it kind of twirls like a ribbon. Okay. Now, in DNA, you're going to have a phosphate, you're going to have a sugar, a nitrogen base, and you'll see these four types of nitrogen bases: adenine, thymine, cytosine, and guanine. So let's say if I drew one of the strands here, and there's adenine for the nitrogen base. On the complementary strand, A will always pair up with T, thymine. If there's guanine, guanine will always pair up with cytosine. So A to T, G to C. It's always that way. So recollect, DNA, they're found within the nucleus in structures called chromosomes. The DNA cannot leave the nucleus. Now, you have instructions to make thousands of proteins, and one particular instruction to make one specific type of protein is called a gene. So you got thousands of different genes to make any little protein you need to make in the body. Now, because DNA cannot leave the nucleus, I have to make a copy of the instructions if I want to make a protein. So the process of making the copy of the instructions is called transcription. Basically, an enzyme will unwind the DNA and make a single-stranded, what's called messenger RNA. The messenger RNA right now has a little bit of junk that needs to be cut off. The junk that gets cut off and stays in the nucleus, those nitrogen bases are referred to as introns. The message itself, when it's finally made and modified, those nitrogen bases that get to leave the nucleus are referred to as exons. They get to exit. Once the messenger RNA gets out of the nucleus, we need to read the instructions to make the protein. So what's attached to the instructions, what you're seeing here is a ribosomal RNA. So ribosomal RNA pretty much provides the building site where we're going to make the protein. And if you recollect, proteins are made up of amino acids. So I'm drawing little spheres representing the amino acids. So as the ribosome goes along the message, you're adding the amino acids to make the protein. And again, the process of making the protein, it occurs outside the nucleus. It's called translation. All right, we'll get to, we don't need, well, basically, look, you're looking at here when you're going to make a protein, not when the cell's copying itself, not when DNA is copying itself. I just want the instructions to make a protein. And one set of instructions to make one protein is called a gene. So what's going to happen, you're going to have an enzyme called RNA polymerase. It's going to unwind the strand and make messenger RNA. And that occurring inside the nucleus is called transcription. So that's what they're showing us here when you're making messenger RNA. All right. Now, typically, when a ribosome attaches to the message, you start making the protein. When it moves along the message and you're adding more amino acids, the first part of the instructions, if they're exposed, another ribosomal RNA can attach to it. So let's just say this was the original one. This was the second one behind it, the third one, and then the fourth one at the start of the message. So anytime the message, the instructions in the beginning is exposed, a new ribosome can attach and make the same protein as well as these others, but at different stages. So that's what, again, this picture is showing. When you get to the end of the message, the ribosome kicks off. There's two parts to the ribosome. You got a large subunit on top, it's larger, and a smaller subunit that kicked off from the bottom. This is, again, an electron micrograph of this in case the messenger RNA with ribosomes attached to it. Sometimes we need to modify the protein by adding sugars, and you recollect the organelle that does that is the rough endoplasmic reticulum. So here you got a messenger RNA outside the nucleus, and you have ribosomes attached, making or providing the building site where we're going to make the protein. Somewhere along the message, it's going to guide the protein to want to go to the rough ER. And at the rough endoplasmic reticulum is where we can add sugars to it, therefore modifying the protein. And then it gets encased in a vesicle, and it's off to the Golgi apparatus, which is the packaging, shipping organelle for protein's final destination. So basically, this picture is just illustrating when proteins are made outside the nucleus, sometimes they need to be modified by adding sugars. And if that's the case, they get taken to the rough endoplasmic reticulum. All right, so in the very top scenario, you're looking at DNA. Remember, a gene is instructions to make one type of protein. And you don't just have one gene, you've got thousands to make any protein in the body. So gene 1 would be instructions for one type of protein, gene 2 instructions for a different type protein, and similar to gene 3 would be instructions to make another type protein. So let's just take one gene into consideration. In one of the strands of DNA, they've shown us the nitrogen bases here. So here you got thymine, adenine, and cytosine. The next ones, you got guanine, thymine. Okay, so you'll have an enzyme unwind the DNA and make messenger RNA. Now, try to recollect A to T, G to C when you're looking at DNA. Okay, so let's say if you had the other strand of DNA here, this T would have bound with an A would have bound with G, guanine. But we're just looking at one of the strands, and we're going to make messenger RNA. You'll never see thymine in the RNA. Notice how you see thymine in DNA, but I never see that nitrogen base thymine in the RNA. You're going to see a different nitrogen base in its place. It's called uracil. So if there's a thymine in DNA for the messenger RNA, you're going to put adenine, A to T. But look here, adenine in the DNA, you're not going to put thymine in the RNA. Instead of thymine, you're going to put down uracil. If there's cytosine, you'll still put down guanine. Then if you're going to read the message outside the nucleus, let's say this is the messenger RNA gets to leave, what's called transfer RNAs. Transfer RNAs are going to bring in the appropriate amino acids to build a protein. So first of all, you're going to need a ribosome to attach to the message. And then the transfer RNAs are going to bring in the amino acids to build the protein. Every 3 nitrogen bases make up instructions to bring in 1 amino acids. So the first 3 nitrogen bases here, you're bringing in this amino acid, I think it's methionine. The next 3 nitrogen bases, those are instructions for a different amino acid, and so forth. So every 3 nitrogen bases, they're referred to as codons in the message. So those are instructions to bring in an amino acid. What's bringing in the amino acid is transfer RNA. How does transfer RNA know it's bringing in the correct amino acid, though? Well, if you see here, A to U will line up, and G to C will line up. Fits perfect, like a key to a lock. So that's how the transfer RNAs know they're bringing in the appropriate amino acid. All right, skip that. We'll come back to that later. We've done mitosis. I want to get to making a protein. Okay, so here we are. We're in the nucleus. Instructions to make proteins are in there. It's DNA, right? You're making the messenger RNA. You'll notice a ribosome is going to attach to the message. And every 3 nitrogen bases are the instructions for 1 amino acid. And what's bringing in the amino acid is called transfer RNA. How does it know it's bringing in the correct amino acid? Again, it has a region here called the anticodon, and it has to fit perfect on the codon region on the messenger RNA. And recollect, the codon is how many nitrogen bases? 1, 2, 3. So this transfer RNA, the anticodon fits perfect to the codon region, and that's how it knows it's bringing in the correct amino acid. What's going on over here, this crescent-shaped protein, that's an enzyme. It's just helping to attach the amino acid to the transfer RNA. So this ribosome is going to go along the message, and as it goes along the codons, will have an accepting site for a new transfer RNA, has the protein building site where the transfer RNA is going to add its amino acid, and then it has an exit site for their transfer RNA to exit from that area. Let's see if I get a larger picture. No, I didn't. So here we go. You got the ribosomes just going along the codons, and each codon are instructions for an amino acid. You put the amino acids together, you're forming the protein. And in chapter 2, this protein happens to be in what's called primary structure, which kind of looks like a rosary with beads, just linear, it's straight. So this ribosome, which is the building site for the protein, is going to go all along the codons of the message until it gets to the last codon, which is a stop message. Hey, you're done. Go ahead and kick off the message, and you finally created your protein, which now is in primary structure. All right, let me jump back. Let's see, what else did I miss? Just to kind of go over it quickly with you. So in this scenario, you're just looking at a pump, protein pump embedded in the cell membrane. All our cell membranes have this pump called the sodium-potassium pump. Basically, it pumps 3 sodiums out of the cell and brings in 2 potassiums. And since you're trying to push the substance from low to high, you're going to need energy in the form of ATP. And this pump establishes what's called a concentration gradient, where you have more sodium on the outside and you have more potassium on the inside of the cell. So that's all it's showing us, that ATP gives up a phosphate to allow this pump to do its job. Sometimes chemicals need to bind to protein receptors before you get the final response at the target. Anything that binds to a protein receptor in the membranes in general is called a ligand. So this chemical ligand bounded to a protein receptor, you go through a cascade of activating proteins where you activate enzymes before you get the final response at the target. This is just showing that little cascade that we saw in this picture earlier. You got a chemical that binds to a receptor, activates proteins to activate enzymes, which activate other proteins called second messengers, and you get a response at the target. We don't need to focus so much in this chapter about this till later. Mitosis, again, what's the purpose? Growth and repair. The stages of mitosis: prophase, metaphase, anaphase, and telophase. Prior to mitosis, where everything's copied, that's called interphase. All right, I'm going to skip all these real quick. Well, no, let's just look. These are larger pictures here. So recollect, cells can copy themselves. Not all cells, but some cells. Prior to copying themselves, where you need growth to occur, you copy the organelles and DNA. That's called interphase. So here you see some centrioles being copied. The DNA is uncondensed still. It's called chromatin. Prophase. How do I know I'm in prophase? That's when the chromatin condenses down to chromosomes. No matter if it's early or late prophase. How do you know in prophase? You see chromosomes. Yes, centrioles are also attaching to the chromosomes, getting ready to separate the sister chromatids. But for me, or until students, you see chromosomes for the first time in prophase. Okay, metaphase, all the chromosomes lined up in the middle to help you that. Remember that, metaphase. Meta means metal. Anaphase, you see now separation of the sister chromatids, or you could say daughter chromosomes. Telophase, you'll see a cleavage furrow forming between the two cells. And last, really not part of mitosis, separating the contents into two cells. It's called cytokinesis. Making a protein, not the same thing as mitosis. The instructions are in the nucleus. You need to make a copy called messenger RNA. Once the messenger RNA gets out of the nucleus, you need to read the message. That's called translation, where you have ribosome involved, which will bind to the message, and transfer RNAs, which will bring in the appropriate amino acids. Ribosomal RNA goes along the message. Every 3 nitrogen bases is called a codon, which are instructions for 1 amino acid. Ribosome goes all along the message till it gets to a stop codon to tell it it's done and kicks off the message. And now your protein is finally produced. It's in primary structure. All right, one last thing I kind of want to show y'all. I'm going to draw one strand of DNA, okay? Just kind of like a ladder. There's adenine, A. T, thymine is another type of nitrogen base. Guanine, and cytosine, okay? Now, if I want DNA, what would the complementary nitrogen base be down here? So if this is DNA, adenine will always pair up with thymine. Here's thymine, so I'm going to put adenine, A to T, and guanine will always pair with cytosine, A to T, G to C. So here's cytosine, and the complementary strand of DNA would be guanine. So for DNA, if you're only giving one strand, they ask us to give the complementary strand. Always remember A to T, G to C. Something to help me remember that is all the good cats. All the A to T. Good cats will always pair up with G and C. Okay? Now, if I want to make messenger RNA, not the complementary strand of DNA like we just did, I want to copy the message to make a protein. So let's do that. When you make messenger RNA, you're never going to see thymine in its place. You're going to see instead uracil. Uh-oh, trying to get the pen out. Let's see here, clear all drawings. Trying to get the pen out. Here we go. All right, so let's do the same scenario. Let's have adenine in the one strand of DNA. Here's thymine. Here's guanine. And then there's cytosine. Okay. Now I want messenger RNA. Messenger RNA is always going to be single-stranded. So if there's adenine in DNA in the messenger RNA, you put M for messenger RNA, you're never going to see thymine. So in the messenger RNA, you're going to see a new nitrogen base called uracil. However, if I see thymine in the DNA, you still can put adenine in the RNA, A. Guanine, you could put a C here. Here's cytosine, you put a G. So when I'm making messenger RNA to help me remember, instead of all the good cats, I would say all usually good cats. A to U, all usually good cats, G to C, when I make RNA. Hopefully that helps. And let me get rid of all these drawings. And I think we've gone over pretty much everything we needed to in this chapter 3. Let's just kind of review what we have here. The movement of substances from high to low, they just naturally want to go where there's more of to where there's less of. You don't need energy. So that type of transport in general is called passive transport. And there's different types of passive transport. So the movement of a substance from high to low concentration, that type of passive transport is called simple diffusion. the movement of a substance from high to low, but it has to be helped across through a membrane by protein channel. That's called facilitated diffusion. And specifically, the movement of water from high to low concentration is referred to as osmosis. One other type of passive transport is called filtration. You probably heard forming a filtrate before. The literal definition of filtration is the formation of a solution from high to low pressure. Since our blood vessels, not just the cell membranes, but our blood vessels are semi-permeable, they'll leak fluid, forming a filtrate, like interstitial fluid. Think of a balloon, has holes in it, and you squeeze the balloon, then liquid's going to leak out, right? From high to low pressure, and you're forming a filtrate. So that's called filtration. That's another type of passive transport. Now, if you're trying to push a substance from low to high, anytime you're trying to push substance low to high, think of like going uphill from low to high. You're trying to push substance where there's less of to where there's more of. You need energy. In this case, in the little analogy I'm giving you, you need energy to get uphill. That in general is called active transport. And what do you need for active transport? You need energy in the form of ATP. And I only need you to know primary active. You need ATP. When a cell takes contents within it, that term's called endocytosis. I need you to focus on the two types of endocytosis. Number one, if the substance large, solid-like, the membrane will surround the contents. That's called phagocytosis. If the content's small, liquid-like, the membrane folds in to bring the contents is pinocytosis. So those are the two types of endocytosis we'll focus on. Anytime a cell excretes something out of it, in general, that's referred to as exocytosis. What is the major structure or component that makes up our cell membranes? Not everything, but the major thing. It's a bilayer of phospholipids. Yes, you can have proteins embedded in the membrane, but the major structure is made up of phospholipids. And then the function of organelles. Mitochondria make ATP. Ribosomal RNA is needed for making a protein. The rough ERs going to modify proteins by adding sugars. The smooth ER is going to break down toxins, also make steroids. Your Golgi apparatus, that's going to be the packaging and shipping of a protein to its final destination. Peroxisomes, they contain enzymes to counteract what's called free radicals. Lysosomes contain digestive enzymes to help break contents inside the cell, should a cell bring contents within it. And then these proteins, microtubules, intermediate filaments, microfilaments, well, in general, you'll see them making up what's called the cytoskeleton of the cell, give it shape, give it structure, to allow organelles to move from one area to another. On the bottom, you got centrioles. Centrioles are to separate chromosomes during mitosis. So they're needed for, well, mitosis. You have cilia. Don't get that confused with microvilli. Cilia is to help propel contents like mucus. They're like hair-like projection of proteins on the cell membrane. Then you have your sperm. Counterpart is the female's egg, contains 23 chromosomes. So that way, when they come together, the sperm and egg, you have a complement of 46 now. Microvilli, those are extensions of the cell membrane to that the cell is going to be better at doing absorption. The nucleus contains DNA, which is instructions to make proteins. If the DNA is uncondensed, it's called chromatin. When it starts to condense, you form chromosomes. The little sphere inside the nucleus is called the nucleolus. That's where our ribosomal RNA and transfer RNAs produce. Contents can leave the nucleus through openings called nuclear pores, which are embedded in the envelope. The nuclear envelope encases or makes the structure of the nucleus. All right, and there again, you see the nucleolus. That's where ribosomal RNA, transfer RNA is produced. and then chromatin can wrap around proteins to form chromosomes. Instructions to make one particular protein is referred to as a gene. All right, we've gone over everything pretty much. That is your chapter 3. I'm going to stop share, and hopefully don't forget coming to lab on Tuesday, right? All right, guys, I'm going to end this recording. I'll take care.